Hello, my name is Rob Bies, and I am the co-director of the Pharmacometric and Clinical Trial Design Core with Dr. Sarah Quinney. I'm very pleased to provide an update on our activities over the last year period. Projects that we undertook included evaluating the disposition of tenofovir in pregnancy, and then this included TAF, TDF, the tenofovir diphosphate levels as well after administration of these compounds. So what we showed, this is a project that was conducted in conjunction with, in collaboration with Impact, Conrad, MTN, HPTN, the University of Washington, the University of Cape Town, among others. We created a model for TAF and TDF that describes the pKal of all the relevant moieties simultaneously, with a particular attention on the disposition in pregnancy. So, according to the semi-mechanistic POP-PK model in pregnancy, we showed that the tenofovir diphosphate concentrations in the peripheral blood mononuclear cells declines 20% with TDF administration in pregnancy. The administration of TAF, however, maintained intracellular tenofovir diphosphate levels, i.e. the active moiety, or the compound responsible for the drug effect, that was 5-fold higher than TDF during the second and third trimesters. It suggests that TAF may provide stable intracellular drug exposure during pregnancy and postpartum. And so the 4 panels on the right are concentration time profiles. And you see the plasma concentrations of non-pregnant second and third trimester, that's the red and the green, and then the postpartum concentrations that are predicted with uncertainty with the between subject variability. And you see that the actual tenofovir plasma concentrations are lower after TAF administration, but the PBMC concentrations are higher. Similarly, the tenofovir plasma concentrations are higher with TDF administration, but the peripheral blood mononuclear cells concentrations, which are the active compound, are lower. We also undertook, in conjunction with the Impact Network, an evaluation of long-acting cabotegravir pharmacokinetics in infants. And this, of course, is an anti-HIV drug. We implemented the ontogeny of the various enzymes in a PBPK model and created a graphical user interface using the R-Shiny open source software. So long-acting injectables provide drug exposure for weeks to months, during which a neonate and infant physiology changes significantly. So we wanted to be able to incorporate those rapid changes over that early period and be able to administer virtual doses to assess expected concentrations after administration of the drug in this population, in particular in the early neonatal period. The Shiny app that's shown, the graphics are shown here on the right, allows the simulation of developmental trajectories and predicts cabotegravir pharmacokinetics in neonates and young infants. We've also been exploring machine learning models and methods in pharmacokinetics. And so we assessed, for FTC, we assessed a multiple objective genetic algorithm, i.e. a machine learning method to select a population pharmacokinetic model that selected that model on 3 key criteria. Those were the objective function values, so how well the model appears to fit the data, the number of total estimated parameters, so how complicated the model is, and the bias in the prediction of the active moiety, the FTC triphosphate concentrations, which are related to the effect of the drug. by doing so, we gained an insight into the trade-offs between competing objectives. So when you optimize for bias, it was able to identify a set of models in which some had less bias than traditional methods. And this can help optimize trial design. So we can evaluate how to determine with the least bias or the greatest precision particular target concentrations and select a model on the basis of that. So this again is using something called the non-dominated sorted genetic algorithm 3, and it's used to solve optimization problems with 3 or more objectives. And in the bottom left table, we see some of the statistics with respect to the range of objective function values on the non-dominated front, the results of the traditional method, the number of estimated parameters in the different models, and the percent of trough PBMC, FTC, TP concentrations in terms of bias. So you can see that this allows the decision maker to choose a model that has a performance on the particular objective of interest. The PCTDC has also been exploring the disposition of antenatal corticosteroids and has implemented a minimal PBPK model and some unique estimation techniques to predict dexamethasone concentrations in pregnancy. A Shiny app has been developed to generate time courses of plasma concentrations of antenatal corticosteroids in the mother and the fetus. And these models are available to use. Dr. Krasansky is the person spearheading this particular effort. Another key project that continues from previous years is a refinement in the understanding of serum creatinine concentrations in neonates. And so a full random effects model was developed to allow for simultaneous use of highly correlated covariates of body weight at birth and gestational age to explain between subject variability of serum creatinine in the neonates. And this was utilized to reduce variability in the prediction of the vancomycin plasma concentrations in neonates with missing serum creatinine concentrations or creatinine observations. So can you use these to help impute or predict in individuals where you may not have the measurement at the exact time, the serum creatinine that allows one to guide the dosing of vancomycin? This is in collaboration with the Catholic University in Leiden. We continue also to refine our models of antibody pharmacokinetics in pediatrics. For monoclonal antibodies with linear pharmacokinetics, neonates and infants have a slightly higher clearance. Pediatrics patients 2 and above have a comparable clearance to adults. So the neonates and infants have the slightly higher clearance, a little bit faster. For monoclonal antibodies with nonlinear pharmacokinetics, pediatric patients had faster elimination unless they were administered saturating doses, at which point they were comparable to adults. Patients under 12 had lower initial concentrations due to higher blood volume and larger distribution volumes. And again, the models underlying these are available to use. And actually, at the end of this presentation, there is a QR code and an HTTP address for access to those models. You can see the changes in the dose normalized C naught here for the different age groups, and changes in the central volume distribution for these moieties. MCRENT, the DMKRCC, and the PCTDC have also been involved in collaborations with the Maternal Fetal Medicine Units Network. We assisted in the considerations of design of a proposed trial to evaluate using oxytocin to induce labor, where the MFMU investigators were interested in evaluating multiple dose levels. The PCTD core will support a KPD, so a kinetic pharmacodynamic model, as concentrations of oxytocin cannot be readily measured. This project, I believe, is under review, peer review at the moment. We also are involved with the MFMU, the University of Utah, in the PregDose study. So this is a proposed NICHD centralized trial with a maternal fetal medicine unit and Kevin Watt at the University of Utah to collect opportunistic samples for concentration measurements to assess changes in pharmacokinetics across gestation. So our knowledge base and real-world evidence cores will prioritize study drug selection. The Pharmacometrics and clinical trial design core will support the PK/PD, the pharmacokinetic and pharmacodynamic study design and analysis. The UCSD Pharmacometrics Analysis Core, led by Jeremiah Momper, will quantify the drugs, and we will jointly develop model-informed precision dosing web apps, very similar to what we have already developed for the antenatal dexamethasone and for the monoclonal antibodies in pediatric patients. The PCTC has also collaborated with Riley Children's Health and used electronic health record data from 449 infants with 2,800 anti-10A samples to develop a population pharmacokinetic model using a Bayesian approach. Age was a covariate on volume and clearance up to 1.5 months, and you can see the postnatal age in the bottom left corner here. This figure, postnatal age on the x-axis and the volume over F term, and you can see how this changes within individuals across that time frame. This allowed one to come up with model-informed doses that would provide plasma anti-10A activity in that therapeutic range, as denoted by these red dashed lines. So you see for less than 1 month, we have most of the patients overlapping with that range. From 1 to 2 months, if you use the typical clinical dose, the simulation showed that we're below the key threshold. The model dose, right, those determined by this model, allowed many more patients to be within this range. If you go more than 2 months, a very similar pattern is observed, with the clinical dose providing subtherapeutic responses, and that the model dose actually getting much closer to the desired therapeutic range. IU School of Medicine is also involved in caffeine and apnea of prematurity assessments, with the hypothesis that administering caffeine prior to delivery will provide therapeutic concentrations at birth. So you have concentration here on the y-axis and time on the x-axis. We have this dosed in utero via the mother, and we see that we then have delivery and dose, and then we're above the therapeutic concentration. If we dose this postnatally, we can maintain those concentrations. Similarly, nifedipine disposition was assessed, and the determination of more optimal dosages considered. So typically 60 milligrams daily dosing leads to a wider peak to trough ratios than 30 milligrams BID. So 30 milligrams BID gives you a much smoother range in terms of, or narrower range in terms of concentration time profiles, and ideally less dramatic swings in blood pressure over the dosage interval in pregnant women. And this is a project in collaboration with Washington University in St. Louis. We have additional ongoing studies. So other studies that were discussed during yesterday's trainee presentations include our FALD, the Fontan-associated liver disease PBPK model, levocetirizine and histamine response in pediatric asthma, labetalol disposition in pregnancy, PBPK equations describing CYP3A and CYP2D6 activity in pregnancy and in postpartum women, population PK studies with the PTN, Pediatric Trials Network, as well as supporting a study of lamotrigine disposition in pregnancy. Finally, I want to provide a QR code here so you can access those monoclonal antibody models. And here's the specific web address down here at the bottom, the link to the application. Many of the completed models are publicly available now as our Shiny apps on the mPrint OSU Azure server. So I thank you for your attention, and I look forward to questions. Thank you very much.